look up there, and the subject is good reading. This stallion won 3 classic races last year, and that makes him worth anything up to £40 million. This mare has a good track record too. There have been no less than 3 national champions in her family tree. The owners of these 2 thoroughbreds have decided to breed from them to create a new champion. The new champion has already been sold to a consortium of businessmen for £15 million, and he's not even born yet. But with his parents, you can afford to be confident. And here he is, Olympic Summer, still a little shaky, but he's finding his feet. A year later, and he's looking good. He's showing promise. 4 members of the consortium, which owns Olympic Summer, have already sold their shares for a large profit. We'll see him again in a year's time for his first race.
And they're off. Bandits Blue Streak leading from Olympic Summer, with Oyster Bay and River Valley half a length behind. Colts put a length behind them, and over on the fence, Georgina and Haymakers showing strongly. An easy win for Oyster Bay, and a good showing for Ray-Ban too. He did better than I expected anyway. But the big surprise is Olympic Summer, fading badly in the last 2 furlongs and sadly adrift in the last straight.
Yeah, it's Jasper. Olympic Summer, I want you to sell him. No, now. I want you to sell him now. Yes. It's useless.
That's first number 1 by Eddie. Second number 4, Solo Act.
Olympic Summer may be a little off colour, but we'll be seeing him again at Newmarket next month, where he may improve. Now it's over to Ray for the 2:30.
Newmarket wasn't much better. Olympic Summer came in 5th out of 6 runners. So what went wrong? With his parentage, why was Olympic Summer such a bad runner? Meet the paramecium. It's a single-celled creature, and it's in solitary confinement on this microscope slide. Come back the next day, and there's quite a few of them, all looking pretty much like the original one. Too bad you can't breed Derby winners that way. But why not? The difference between the paramecium and the horse is that it takes a mare and a stallion to produce a foal. But when the paramecium reproduces, it simply splits in half, producing 2 identical new paramecia. Why can't racehorses do that? To answer this question, we have to consider the 2 different basic types of reproduction. With asexual reproduction, as we saw with the paramecium, there is only one parent. The offspring inherit all their genetic information from that parent and are exact copies of it. Sexual reproduction, as we saw with Olympic Summer, is very different. There are 2 parents. The offspring inherit genetic information from both parents. But where would you find this genetic information? Well, inside each cell is the small round nucleus. Inside the nucleus are the chromosomes, strands of DNA. They determine whether the cell is going to be a cat or a camera operator. The paramecium reproduces asexually. Each cell has paired sets of chromosomes. This is called diploid. First, the chromosomes duplicate themselves. An identical paired set is transferred to each of the daughter cells. This form of cell division is called mitosis. Asexual reproduction is found in plants too. This strawberry plant has sent out a runner with a small plantlet growing at the end. The runner will break down and leave a new plant identical to its single parent. There's a great deal of money to be made out of this. Here's a tray of young African violets and a particularly good adult specimen. How does the commercial nursery coax the prize plant into producing new plants of the same high quality? First, they chop off its leaves. Then they soak the leaves in disinfectant solution. But they don't stop there. In the next stage of the process, they cut the leaves into small pieces. Each piece is composed of genetically identical cells. Now, what do you think would happen if we took those pieces of leaf, sealed them up in tubs of growth medium, and then stored them under controlled conditions for 2 or 3 weeks? They grow into a host of genetically identical plantlets. They may even look identical to each other, as well as to their parent. Asexual reproduction, as we saw in the paramecium, generates uniformity. These small green insects are aphids, a real menace to the farmer and the gardener, as they appear in sudden swarms and infest the crops and rose bushes. Every insect in the swarm is likely to be genetically identical. In fact, the entire swarm may have come from a single aphid. In spring and summer, when there is plenty of food available for the aphid, it can increase its numbers incredibly rapidly. They don't need to mate. They reproduce asexually by a method known as parthenogenesis, or virgin birth. Here's another aphid giving birth by this method. Aphids breed so rapidly that in one summer they could cover the world with a layer of aphids 1 kilometre deep. So, why doesn't that happen? We've seen how efficient asexual reproduction can be. Now we'll change the aphid's environment. We'll decrease the day length so they think it's autumn. How do you think they'll respond? Amazingly, some of the baby aphids grow into males. They're the ones with the wings, and the species switches to sexual reproduction. Here are a male and female aphid mating. The male sperm and the female ovum will join together. Both parents will hand on characteristics to their offspring. They will hatch out from these eggs in the next spring when the days are longer. We'll go back to horses for a closer look at sexual reproduction. Like the aphids, horses are diploid. Let's say their body cells contain 2 pairs of chromosomes. When a male sex cell, or sperm, fertilizes a female sex cell, or egg, the foal will have 4 pairs of chromosomes. Its offspring will have 8 pairs of chromosomes. The generation after that will have 16 pairs. Well, that can't be right. So how's it done? Sex cells, such as these sperm cells, are not diploid, but haploid. They contain only 1 set of chromosomes, exactly half of the genetic material found in the body cells. To produce them, diploid cells must undergo reduction division, or meiosis. The nucleus fades and the chromosomes appear. The pairs of chromosomes come together. Then, each chromosome divides in 2. the double chromosomes are pulled apart to opposite ends of the cell. Then the double chromosomes are also pulled apart, leaving 4 sex cells, each with a single set of chromosomes. When a sperm and ovum join, the fertilized egg has, once again, 2 sets of chromosomes, carrying genetic information from both parents. The long dark yellow structures at the centre of this tiger lily are anthers. They produce pollen. Bees pick up pollen from the anthers of one plant and transfer it to the stigma of another of the same species. From there, the pollen transfers its genetic material to the ovule or female sex cell at the base of the stigma in the ovary. To do this, the pollen grows a pollen tube, and through this sends its chromosomes down to join with the chromosomes of the waiting ovule. The new seed will show characteristics of both parent plants. It all seems a very complex process, so what do we need sex for? What do we need sex for? Well, there are several answers to this trick question. If human reproduction was asexual, you would look exactly like your parent, and so would everyone else. There is one problem. Asexual reproduction means that all these plants are genetically identical, which can be very desirable, but it can mean that a pathogen which kills one plant may kill the entire crop. Genetic diversity, on the other hand, means that some might survive. Sexual reproduction is a very effective way of creating genetic diversity, which is why Olympic Summer wasn't a carbon copy of his parents. The breeders were unlucky. Instead of inheriting the best characteristics from his parents, he inherited some of their less desirable features. Sexual reproduction generates diversity, but at the same time, it's a gamble. And that's what the science of genetics is all about, improving the odds. Take one white mouse and one black mouse, one of each sex. Leave to simmer a couple of weeks, and here's one we prepared earlier, a litter of young mice. At this stage, they only come in one colour, pink. But what colour will they be when they grow up? And here they are, 2 months later, a box of black mice. Why aren't there any white mice? We'll take 2 of these black mice and breed from them. What colour do you think their litter is going to be? Here's a surprise. In a litter of 4 mice, there's one white one. Just like the grandparent. It's as if the white fur skipped a generation. Confused? Information held at the same point on each of a pair of chromosomes controls the same characteristics. We call these locations genes. The different types of information carried by the genes are called the alleles of the gene. This cell has one allele for black hair colour and one for white hair colour. Remember the first mice? The black mouse had 2 alleles for black fur and the white mouse had 2 alleles for white fur. So when they mated, each resulting mouse had 1 allele for black fur and 1 allele for white fur. The mice look black because the allele for black fur is dominant. It masks the effect of the allele for white fur, which is recessive, but it can be passed on to the next generation. When these mice produced sex cells, half of them carried the black allele and half carried the white allele. So when they mated, one young mouse had 2 black alleles. 2 mice had 1 of each colour, and 1 mouse had 2 white alleles. This was the one white mouse which reappeared unexpectedly in the last generation of mice. Now we know why. Here are the grandparents, the parents and the youngsters. The white fur colour could only reappear in the last generation in the one mouse, which, like its grandparent, had two alleles for white fur. But it's not always as clear-cut. With many alleles, there is no dominant or recessive feature. In normal human blood, the red blood cells are smooth and round. They flow easily through our arteries and veins. A single genetic change, however, can cause sickle cell anemia. Sickle cells tend to stick together in the bloodstream, causing an eventually fatal illness. People with the sickle cell gene would not be expected to have many children, and the gene would gradually die out. But in certain parts of the world, it is very common. Far from dying out, the frequency of this bad gene has remained constant since it was first mapped. Why should the gene have remained in these regions and nowhere else? Oddly enough, in some parts of the world, the map of sickle cell genes overlaps almost exactly with the distribution of malaria. In these countries, it's an advantage to have the sickle cell gene. It gives a resistance to the malaria parasite, so a bad gene becomes a good one, or at least not entirely bad. The story of the way in which wheat has been bred is another example of how difficult it is to define a good gene. These tall wheats were grown over 100 years ago. What were they wanted for? Straw hats. The hats went out of fashion, but the wheat is still grown for the long straw used in thatching. Short straw cereals are better at carrying the heavy seed heads of the modern high-yielding varieties and are more suitable for cutting with modern machinery. Then we come to the seeds themselves. You might want one type if the wheat is going to be milled for bread flour, another type for pasta, and a third for feeding livestock. We are no longer talking about a good gene. Instead, we have moved on to thinking in terms of suitability for a specific purpose. The desired characteristics depend on the market and the environment. What does the market want? What is the soil type? What are the local grain pests and diseases? And what is the temperature throughout the year? The quest for the right plant is an endless one. Very high-yielding varieties might be well suited to a country able to afford the nitrate fertilizer such strains need. But there would be little point in exporting these same strains to a poor country unable to afford such fertilizer. One way to overcome this problem is to try to get the plant to provide its own fertilizer. Some plants can do this naturally. Peas and beans have nodules on their roots, full of nitrogen-fixing bacteria. These supply the plant with all the nitrogen they need. Scientists have tried to isolate the genes responsible for producing these nodules and to insert them into the genetic code of wheat. The ultimate aim is new varieties of wheat which need no fertilizer and which can grow in the poorest of soils. Could human beings be improved in this way?